This episode of Cheat Codes, a Sickle Cell podcast, is intended for educational and informational purposes only.
What's going on, Warriors? Welcome to another episode of Cheat Codes. This is a special episode because this is actually not a standard Cheat Codes episode. We welcome you officially to What Just Happened, the Best of Sickle Cell Conferences. Dr. Callaghan, this is something we've been wanting to do for a little while.
For sure. And this is really a special episode. I mean, it's a little different than our normal episode, but we got a special group of people on today.
Man, I agree. And I always talk about how, you know, the era of innovation we're in, this period is what they're going to write about 10 years from now, 15 years from now, about when sickle cell disease changed. We're living it.
Hope it's going to be a great story.
All right, so let's jump into some intros because we've got an all-star cast, man. We assembled the Avengers for this. And I'm going to kick it to somebody who really needs no introduction in the sickle cell community, Dr. Lakiea Bailey. Dr. Bailey, welcome.
Thank you. I'm so glad to be here. Thank you for assembling this awesome team. I am Lakiea Bailey, and I am the Executive Director of the Sickle Cell Community Consortium. And I am excited to co-host this with you all for this amazing topic.
Awesome. We're so excited to have you, Dr. Bailey. Let's go to our next caregiver moderator, Ray Blaylark, also no stranger to the sickle cell community. Ray, welcome, welcome, welcome.
Thank you. I am happy to be here. Yes, I am Ray Blaylark, and I am the caregiver or co-care provider to my 24-year-old son, who is a warrior living with sickle cell disease. I'm extremely happy to be here. It's cold outside, but it's warm in here.
Glad to have you. All right, the Godfather, Dr. Wally Smith, a Cheat Codes alumnus. Welcome back, Dr. Smith. We're glad to have you.
What's up, Warriors?
I love it. All right. That's it, right? The Godfather doesn't need to say more than that. We're good. Let's go next to Mr. Worldwide, Dr. Jeremie Estepp, another Cheat Codes alumnus. Dr. Jeremie, welcome.
Hello, everyone. It's so great to be back with you guys, Dr. Z and Dr. C, and all of the illustrious Avengers today. This is a great time to get together, and it's certainly fun to talk about this amazing science.
Thank you, Jeremie. Last but not least, another sort of big-time Avenger here in the sickle cell landscape, Dr. Drew Campbell. Drew, welcome.
Thank you, everyone, for allowing me to participate in this, I think, great discussion. Great to see everyone. Again, I'm happy to contribute to the knowledge in the community. I'm humbled. Thank you so much.
Awesome. All right. We have a lot to cover. We're going to have to run through this, and we're going to have to full court press the whole time. So everybody be ready to sweat and hustle a little bit to get this knowledge out to the Warriors. I'm going to pass it, though, to Dr. Bailey, because I want Dr. Bailey to introduce the concept a little bit here of why we actually have come together to do this and what we're hoping to accomplish.
Thank you. And I love this concept. We were, Dr. Z, you and I, we were sort of on the same page, but in different areas, wanting to do something very similar, where there are amazing sickle cell conferences that occur around the world, and there's fantastic science that comes from it, but patients never hear that. And what we are missing is an opportunity for physicians, for scientists, researchers, patients, caregivers to sit around and talk about what just happened. What did I just hear? Did you see that amazing science? And so we are excited to be sponsored by Agios in order to present this podcast as part of our Citizen Scientist, Unpacking the Science of Sickle Cell initiative that we are unrolling for 2021. So we're glad to be brought together to talk about this and to talk about all the cool science at our most recent meeting, the first that we're covering, ASH of 2020.
I love it. Thank you, Dr. Bailey, for that. So with that, let's jump right into it, guys. And I thought that we could kick off this session of what just happened with starting by talking about the area that's really hot and buzzing right now, which is curative therapy, right? We know there's a lot going on. This is obviously an area of immense attention currently. The rigor of science and the way clinical trials are done in order to protect people is showing itself, right? We're showing how we go through clinical trials in such a rigorous fashion. But I think some of the data that was presented at ASH is really important to share with the warrior community. And for that, I'm going to pass it over to Dr. Callaghan first to introduce the first abstract of discussion.
Yeah, thanks, Dr. Z. So we're going to talk today about a number of presentations from the recent American Society of Hematology meeting. And this is sort of our Super Bowl in hematology. I mean, we have 25,000 researchers, doctors, healthcare providers, advocates, everybody from all around the world join in this meeting. And it's often where we see the best science come out. It's the biggest stage. And so the people who are putting out science want to have it shown on that big stage. So we're going to talk today about some curative therapies. We're going to talk about some pipeline drugs. We're going to talk about some advances with therapies we already have. I think it should be said, though, this is all research. So a lot of it is things that are going to be important in the future, hopefully, but they might not be available now, or if they are, they're through clinical trials. And as with everything we talk about here, this is for education. It's to provide the warriors information, but decisions about your medical care you should make with your doctor. So hopefully you learn some interesting things here that you go talk to your doctor about in your home team, your medical team. So I'm going to kick it off talking about curative therapies. And I think Dr. Campbell and I are going to talk about three great presentations from the ASH meeting. But I want to take a step back and talk about what are curative therapies. So right now we already have some approved curative therapies, and most of them involve replacing the stem cells in our bone marrow. So we have stem cells in our bone marrow that grow baby red blood cells that turn into the red blood cells that circulate in our blood. And if you have sickle cell, those make sickle hemoglobin and you have sickling. So there's a lot of treatments that replace those stem cells. Some of them are bone marrow transplant, and we might do a bone marrow transplant where we take your sibling's bone marrow, your brother or sister, and use their bone marrow to replace yours. So you make non-sickle cells. Now they're doing some things called haploidentical, where you might be able to use somebody who half matches you, like one of your parents, or even unrelated transplants. But one problem with all of these is you need to find a match. The match is never perfect, so it can always attack your body a little bit, and you can have something called graft versus host disease. And most of our warriors don't have a good match, so they're not eligible for these therapies. With that in mind, they've created these new therapies where they take your own stem cells out and then manipulate them. They use usually a virus to put a gene in or edit some of your genes so that your cells make either variant hemoglobin or fetal hemoglobin, so that it prevents sickling. The first study I'm going to talk about today is one of these that uses a really clever new technique to edit these stem cells so that they make fetal hemoglobin. So this was a study from a company called Crispr Therapeutics and Vertex, who helped sponsor the study. And it was called Safety and Efficacy of CTX001. So that's the new therapy in patients with transfusion-dependent beta thalassemia and sickle cell anemia. Early results from the CLIMB Thal 111 trial and CLIMB SCD 121 studies of CRISPR-Cas9 modified CD34 hematopoietic stem and progenitor cells. So that's a mouthful, but we're going to break it down. So as a little bit of background, when we're babies in utero, we make fetal hemoglobin, and that hemoglobin doesn't sickle. But when we're born, we switch to our adult hemoglobin. And if you have the sickle cell mutation, then that's when you start to have sickling. There's a group of people who have a mutation in a thing called BCL11 that's involved in that switch. It turns on a switch so that you stop making the fetal and start making the adult hemoglobin. There's a group of people that have a mutation in that, and their switch doesn't turn. And so their cells continue to make fetal hemoglobin even after they're older. And that can be protective in sickle cell. That's why we use hydroxyurea, and we know people who make more fetal hemoglobin are more protected. So in this study, what they tried to do is take the stem cells out and use this thing called CRISPR. And CRISPR has been relatively new. There are things that are found in bacteria and help bacteria not get infected by viruses. And what they are is clustered, regular, interspaced, short palindromic repeats. But that's too much for me. So I just think of it as like a little template that sticks to a specific spot in your DNA. And then attached to it is a Cas9, which I think of as like a pair of scissors. So that CRISPR goes in, it binds to the specific spot in your DNA, the scissors cut it and make a mutation there. And in this case, the CRISPR goes to this BCL11, and then the scissors cut it, and then you have this mutation like the people who make fetal hemoglobin. So this study is a phase 1 study, which is an early study to see if something is safe and possible, really. And what they did is they used a lentivirus, which is a kind of virus that can infect people. And it's a special virus because it takes the DNA from the virus and puts it into your DNA. And then that makes this CRISPR Cas9 that then goes and changes your cells, changes the DNA in BCL11. And then they do basically the same thing we do with a bone marrow transplant. You get a preparatory regimen that contains busulfan, which is a chemotherapy to get rid of your own stem cells. And then they give you back your stem cells that now have that change. So in this study, they enrolled people who were 18 to 35 years old who had thalassemia or sickle cell, and they had to have at least 2 pain episodes a year. And then they gave them some medicine called plerixafor to make their stem cells come out. And they used a machine called pheresis that you might use if you're getting blood transfusions to collect the stem cells. Then the company used this CRISPR, the lentivirus and the CRISPR to change those stem cells. And then the patients were admitted and got this bone marrow transplant with their own cells back. So there were 2 sickle cell patients on this study. So this is a really early study. They had 7 and 7.5 pain episodes a year over the previous couple of years. There are also 5 thalassemia patients. And in the thalassemia patients, they don't make hemoglobin well. So they wanted to see if by switching them to the fetal hemoglobin, they could make hemoglobin and not need transfusions. It's early days, but this went really well. So all of the patients are at least 3 months out. Some of them are about 1.5 years out. And so they all underwent this procedure. It engrafted. So that means the new stem cells came in, they took up residence and started making cells. And that took about 4 weeks, 3 or 4 weeks for both of these. But the punchline is that after up to 15 months in the thalassemia patients, they're not needing transfusions. They have high hemoglobins. And in the sickle cell patients, about half of their hemoglobin is fetal hemoglobin now, and they're not having pain episodes. So this was a really exciting presentation at the ASH meeting. They pick about 6 of the best out of tens of thousands of presentations that they say are the plenaries. They're the best ones. This was one of those. And I think it really is exciting science and really has the potential to help a lot of people. So with that, I'll open it up to this panel for discussion, and can check me on big words I used and clear anything up that wasn't clear.
You talked about so many things, and it's such an interesting science, but you hit on something that I've often wondered about, and that is you kind of talked about persistent high fetal hemoglobin, as it's commonly known. And when you talk about that, you also mentioned that they typically don't have as many crises or as many complications, and yet sometimes people who were told that they had persistent high fetal hemoglobin in their childhood, we see them almost catching up to those who oftentimes have more crises. Can you explain how that would impact a study like this?
Yeah, I think that's a really important point. So there's a lot of variability in fetal hemoglobin. So some people make just a little bit, some people make more. And we've seen that the more you make, probably the less symptoms of sickle cell you might have. And we use fetal hemoglobin for that reason, to try to increase it. There's a really small group of people who have really high levels of fetal hemoglobin. They maybe have 30, 40, 50%. Some of them from different genetic variations they have. And they are protected from a lot of the complications of sickle cell. But as you mentioned, these aren't magic bullets. They don't prevent everything. And there's still some sickle cells and sickling going on, and over time that can cause damage. So as you said, as people get into their older years, that damage accumulates. I think Dr. Smith says, the hum of sickle cell starts when you're born and accumulates over time. And I think this is an example that, having a high fetal hemoglobin protects you some, but not completely. I think it protects you more if it's in a lot of cells. So sometimes you have high fetal hemoglobin in one group of cells, but not in another. And the ones that don't have high fetal hemoglobin are sickling and causing problems. So one thing that's really important is to look at, how many of your cells have fetal hemoglobin in them. And that's something they looked at on this study and on a lot of the other gene therapy studies. And I think there is some variation there, but in this study, it looks like most of the cells have a lot of fetal hemoglobin. So I think that's promising. I think, like you said, though, the effects long-term are going to be important, and we don't know. I mean, we have people are about 15 months out now. How long is this going to last? Are we still going to see sickle cell related problems, hopefully to a much lesser extent, but will we see them? I think all of those things are, why we do these studies and why we need to follow people for a really long time afterwards.
Excellent, excellent. And then you also talked about that the qualifications for participating in the study was if you had had 2 or more pain episodes in the past year. Now we know once you hit, that young adulthood, you really you're not in the hospital. You look for ways to avoid having to be in the hospital. So was that hospitalizations or was that self-reported?
That's a good question. And in this study, it was pain episodes that required medical utilization. So these were people who were admitted to the hospital more than 2 times a year. And in the 2 patients who were on the study, one of them was admitted 14 times in the previous 2 years, one 15 times. So they had a lot of pain episodes. I think, the principle of this would work in people who don't have pain episodes, but always on these studies, we have this principle we call equipoise, which is, there's always some risk to a study, and we don't want to put people at risk who might not benefit from it. So if you're not having pain episodes and you hope that this new therapy is going to improve pain episodes, you don't get that benefit. But if you take a patient on the study who has a lot of pain episodes, then they could potentially get some benefit from it. And hopefully that balances with the risk.
I was just going to give you guys a little bit of insight. ASH was virtual, but this is an abstract that caused a lot of buzz. People were buzzing about this one. This is the one that got press attention. This is the one that people were talking about. So, being a hematologist, watching this unfold in the sickle cell space, that feeling was unbelievable. And I really wish we got to experience that in person, but we didn't. And I think we had a lot of conversations internally as sickle cell physicians, right? So certainly me and Dr. Mike did, but I'm curious just to hear from, Jeremie, Wally, Drew, you guys, when you first saw these data, what are you thinking?
So, Mike, you're mucking around with my DNA. Is that safe?
Yeah, I mean, that's
Valid.
the question of the study, right? This CRISPR technology is pretty new. One concern is that, we think that the CRISPR goes to this one spot in your DNA and makes a cut. Could it accidentally go to other spots and make cuts that don't help you? That's a risk with these things. And I think, that's why we're doing these studies, to see if it's safe. Now, I should say, things don't just jump into phase 1 clinical trials in people. There's always a lot of preclinical work. There's work in cells, in culture. There's work in animals. And we don't jump into human trials until we think there's a prospect that it can be done safely. But until you do it in people aren't mice, people aren't dogs. We see different results when you try it in people. And I think it's going to be really important that we track these studies really closely and make sure that they continue to be safe before we start doing it as a clinical.
And isn't it true that they're going to follow people for like 15 years or something like that to make sure it's safe?
It's
So scientists, can we take a minute and for our patients and caregivers who are not science nerds, take a minute and let's take a step back. And CRISPR, so we're talking about CRISPR, which is a type of gene therapy. And specifically what made this ooey wow is this technology, when it first came out several years ago now, it provides a way it manipulates something that's seen in nature where microbes can capture snippets of the invader DNA and then target and destroy it. And what this does is it uses that Cas9 to find those little snippets, and it can guide it directly to the right spot to make the cuts. And so this is a way of guiding exactly where you want to make that change in the DNA. And so can you talk a little bit about why that was such a cool and revolutionary, and what it is, how that separates it from some other types of gene therapy that we see?
Yeah, I mean, this really was a big advance. They won the Nobel Prize last year, actually, for discovering these CRISPRs. And I think it's an amazing tool. It allows you to really efficiently. So we had other tools that could go in and do this kind of thing, but they couldn't do it repeatedly. They couldn't do it across all the cells. So this one was a lot more efficient, and they're also a lot easier to make. So there are some other technologies, and some of them are being used like this. And the other thing that's, a potential for this is you could go in and edit. You could go in and cut where you want to, and then you could fix where you cut. So we call that edit repair. So for instance, sickle cell, everybody has the same mutation. You could go in and cut out that mutation and then repair it. We're not quite there yet, but it's really exciting. A lot of the other gene therapies, and I think Drew's going to talk about a couple of them in a second, are doing something we call gene addition. And this is a little cruder. You're just using the virus to put in a hemoglobin gene. It's still amazing, but it's a little bit cruder. And so this CRISPR allows us to do some, I think really elegant things like go in and edit BCL and increase fetal hemoglobin.
As Dr. Smith was talking about, this is, one of the concerns I think with all these gene therapies is kind of we call off-target effects, and Dr. Callaghan talked about that. So we're excited about the science and technology, but safety is very important. And so one of the things that, the reason we want to look at this over so many years is this is novel, this is experimental, but, it's very limited experience in using this for any type of human disease. So we have to, over time, just make sure that the CRISPR is keeping things in check. Your body's keeping things in check with the CRISPR. There's no long-term side effects as this is kind of replication, if you will, in your stem cells. So this is revolutionary. So as this is happening over time, we have to follow everything, the side effects, the course of the sickle cell disease. So this is not a short term, this is a long-term process. And then we'll talk about this in the other types of gene therapy.
Since we're talking about all of these novel or new sciences, is all of that protectionist, how does that fit into medical ethics?
Yeah, that's a great question. And I think Dr. Estepp was on Cheat Codes episode 16, and we spent a really long time having a discussion about Tuskegee and about what an institutional review board does, and really all of the elements that we try to put in place to make sure that we're doing a study that can answer the question we want, but also do it in the safest way possible, in the most ethical way possible, that we're choosing patients for the study fairly, that we're not excluding people, we're not, using a vulnerable population as guinea pigs, and really try to do these things in the safest, most ethical and effective way possible. So I would say, if you want a whole lot more depth on that, episode 16 of Cheat Codes.
Excellent. And I think it feels like the most important thing is that this is not, ethics has evolved. This is not the history that was present many, many years ago, but there are actually laws in place that help to protect those who participate.
And that's a really good segue to Drew's next abstract. So we know that those laws that were in place, the rigor of science, has obviously resulted in some changes to the gene therapy scene as we know it. Drew, you want to take us into that discussion a little bit?
Of course. So first thing I want to say is that, a lot of these results that we're talking about are as of, like, for example, the Bluebird Bio clinical trial was reported as of March 2020, and the Aravant gene therapy trial that was presented as of July 2020. So that's very important, especially in present day, because obviously there's been developments with the lentivirus gene therapy. So let's talk about this. I think we had a nice introduction from Dr. Callaghan. So the first gene therapy abstract I'm going to talk about is the bluebird bio one. This was presented. The title was, results from the ongoing phase 1/2 hemoglobin 206 group C, just the name of the trial study of the lentiglobin for sickle cell gene therapy. Unlike what Dr. Callaghan was talking about with the Crispr, this is the gene addition, meaning that they're actually using a virus called the lentivirus, similar to the HIV virus, where they remove the HIV-like proteins out of the lentivirus, and they actually put in elements of this gene addition. So it allows this lentivirus, if you will, to incorporate a new hemoglobin, not the sickle cell hemoglobin, but a normal-like hemoglobin. So the goal is to increase the amount of non-sickle hemoglobin, which we'll talk about, that will decrease sickle cell related transfusions, complications, and of course, most importantly, clinically, less pain crisis, less organ damage over time. So so in this study, they included patients 12 to 50 years of age with a history of sickle cell, of course, stroke or severe vaso-occlusive events. And what they did is they looked at either patients with increased pain episodes or acute chest syndrome. Also, in this trial, they monitored for our usual blood cell count, also markers of hemolysis. So like for your reticulocyte count, and you know that we also measure things like LDH and bilirubin that's released when your sickle cells don't last that long, and also pain. And then also they reported on patient-reported outcomes, which essentially is kind of the survey questions from the PROMIS questionnaire. We won't go into detail, but to see how much pain you have during different parts of the study, different times of the study, and of course, adverse events. So essentially, in this group C of the trial, I won't go into detail, but essentially out of the 40 patients, they reported on 25 out of the 40 that were treated with this lentivirus gene therapy for a median. Median is another way of doing average, for a median of 12 months, so about a year. It ranged from 2.8 months up into 24.8 months. So the results were the following. So they reported on, well, how well is the lentivirus transplant, how well is it working, right? So again, they're taking the patient's stem cells, right? And then they're using the lentivirus outside of the body to infect the patient's stem cells. And then they infuse it back into the sickle cell patient. Okay, so they actually, for just any bone marrow transplant, they want to determine how well white blood cells, red blood cells, and platelets actually we call engraft. And what we mean by engrafting, we mean that how high the white blood cell it decreases to almost zero and it comes right back up. So they measure the number of white cells you have, and also platelets. Platelets helps with bleeding, so that drops down, and then they measure after transplant how much time it takes for it to go up. So the neutrophil engraftment, the time for engraftment was 19 days, the median time, average time, and the platelets was 28 days. But more importantly, how does it relate to sickle cell disease? So in all the patients, they stopped blood transfusions by 90 days post-transplant. So basically, after the lentivirus treatment of each patient, so 90 days, nobody needed any blood transfusion. In 16 patients that they could follow, that was followed up for greater than 6 months, okay, the average hemoglobin was 11.5 for the median. The range was 9.6 to 16, so as high as 16. It's pretty impressive. And the transferred gene hemoglobin, which they called this gene addition hemoglobin, is called T87Q. You'll hear me say that, right? This T87Q contribution of the hemoglobin was about half of what the total hemoglobin. So the average hemoglobin was 11.5, right? The gene addition hemoglobin contributed an average of 5.2. And the sickle cell hemoglobin contributed to a hemoglobin of 6.1. So it's about half. So you almost have half and half. The other thing they did to make sure that the gene therapy was truly expressed in these red blood cells of sickle cell patients, they did these we call assays. So outside of the lab, they took the patient's blood cells, and they measured how well that this gene therapy, this gene addition hemoglobin 787Q, how much of it was expressed. And they found that it was we call pan expression. So in almost all the red blood cells, their assay showed that this gene therapy addition hemoglobin was expressed in almost all the red blood cells in 9 patients who had this gene therapy greater than 6 months. Okay, and then 90% of the red blood cells had some level of this T87Q hemoglobin by 18 months. And then they looked at this hemolysis markers, which we talked about. Sickle cells, sickle cells only last 20 to 30 days. So there's increased turnover of red blood cells. So you have an increase in hemolysis, right? So after the transplant, after the gene therapy, they show normalization of LDH, reticulocytes, and bilirubin. So in terms of the most important outcomes was pain crisis, right? So they broke it down into vaso-occlusive crisis, which is painful episodes, and acute chest syndrome. Okay, so then what they did was they actually added them. So prior to treatment, there was a average, we call annualized, so for every year, vaso-occlusive and acute chest syndrome rate total of 4 in the 2 years prior to treatment. So every year, there was at least 4 episodes of pain, crisis, or acute chest syndrome. Post-treatment, that number went down to 2. So, and in fact, there was, based on the results, they said there was a 99.5% mean reduction in annualized vaso-occlusive and acute chest syndrome rates post-transplant. And they showed this really nice figure showing that. So essentially what they showed with this, in addition to that, they also found that the PROMIS scores, these pain intensity scores, also was reduced 12 months post-treatment in 5 patients who said that they had pain intensity scores that were worse prior to going to transplant. And then another 5 patients said, going to transplant, their scores were near or better than the population, was about the same. So there was a half of them said there was improvement. The other half said it stayed better. Okay. And then the severe adverse events, things have changed now, but at the time, right, the most common, side effect was just secondary to the we call preparative regimen, which I kind of glossed over initially because I know we'll talk about this, is that as part of this treatment, everyone gets a little bit of chemotherapy so the transplant can work. They get what's called busulfan. So as part of that, the reason they give the busulfan to the patient prior to getting the gene therapy is because the belief is that sickle cell patients' bone marrow is a little inflamed, even getting a self-transplant. So they cool it down by giving some both busulfan, which is the chemotherapy regimen they use for transplant patients, to cool down the inflammation, so they can accept their stem cells that have the gene therapy in it, right? So with that, when you get chemotherapy, you get stomatitis, we call it, with inflammation inside the mouth, and also you can get neutropenia with the white cells dropping down. So that was the most common, we call grade 3 and above adverse events. That was non-hematologic. The other serious events were very small, and that those severe and those neutropenia, stomatitis, that was in 11 patients, and then nausea, opioid withdrawal, and vomiting were just in 2. 3 patients had lentiglobin virus, non-related grade, less than grade 2 adverse events. These are just different gradings. And then there was 1 death They thought at the time that was unrelated to the lentivirus. It was greater than 18 months post-treatment, which we know happened in July of 2020, post-treatment in a patient with significant baseline sickle cell-related cardiopulmonary disease. There have been no events of graft failure, vector-related replication competent lentivirus, or clonal dominance. So we know that has changed. So basically in this trial, this study, they said that the lentiglobin virus gene therapy resulted in pancellular, almost all the red blood cells expression, and reduced sickle cell expression, okay? That's decreased the complications that you would see with sickle cell disease, but also they described decreased, which had an impact on the pathophysiology or the impact on sickle cell disease by reducing sickle cell sickling and reducing hemolysis, but increasing total hemoglobin. And with this, they also said complete resolution of vaso-occlusive crisis and acute chest syndrome was observed in almost all patients, with a 99.5% reduction in annualized for every year of vaso-occlusive and acute chest syndrome post-treatment. And there was also a decrease in reported pain score intensity. So the bottom line is, from our standpoint, it was very exciting. We were very excited about this trial, okay? Now, we know that there's been changes that's happened recently, which I will pause right now because I know that can be another discussion.
The trials have been so amazing, and there's fantastic information that comes out of both of it. You said one thing that I know we're going to get, and I'm sure Ray can attest to, that we're going to get and we're going to hear again. And so often it gets glazed right over, and it is simply HIV-like lentivirus. And so sort of one of the reasons that HIV was such a difficult disease and such a hard one to get was because it is so good at injecting its virus into cells. But that thing that made it difficult to treat and just a nightmare of a disease is the same thing that becomes useful in this, because the virus is removed. And now we've got this vessel that is fantastic at getting what we put in it into a cell. And so when we talk about that, just to clarify, because it has come up repeatedly, this is not an AIDS causing thing. This is not going to cause AIDS and HIV. It's simply that it's using that technique that is so good at getting its message into the cells. So I want to say it, just to pause right there to just clarify that to those listening. We have so many wonderful things that we want to talk about. So I wanted to just get a really brief description. We won't be able to go into all of the details with the next trial, but I wanted to hear some of your thoughts about that. Some of what were your initial reactions to this study, and Ray, your comments as well.
Yeah, so I'm glad that you brought that up because, sometimes we can hear certain words that feel triggering, right? And we're not sure. There's a lot of history behind there. So has that been used before? Do we understand if lentiviruses have a history in science? Or is this the first time and this is new and they just decided sickle cell disease was going to be the disease they try it out on?
It's not new. The lentiviral platform has been used in multiple other diseases such as hemophilia B, X-linked skids, macular degeneration. Mm-hmm. So it wasn't that sickle cell disease patients were selected to have this platform tried on them. It was that this platform is just exceptionally powerful with respect to being able to insert genes and to change information. I use the analogy in clinic all the time of it's like taking an old beat up pickup truck that is very useful and dropping a V8 engine in it because it changes the way that it functions. It just becomes very much useful.
So no one's ever got HIV from using this science?
No.
No.
And I think it's important to just point out that there's a long history to this. We often hear that our community will think, well, we're being used as guinea pigs. And there is actually a very long history to this, and it did not start in sickle cell. It started in other illnesses, and even the way it's being used now was done first in thalassemia. And so I think it's important to talk about that. Sickle cell is a fantastic model illness, right, because we know exactly its molecular basis. And so we can then use that. And sickle cell has actually been connected to a number of scientific advances, including a previous Nobel Prize in chemistry. Because we understand how it works, we can then use that in other ways. But Dr. Campbell, I'm wondering if you could rather quickly compare this to Aruvant.
And Dr. Drew, could you give us like a super high level difference on what's the difference between bluebird and Aruvant? That'd be great.
So the difference between the Aruvant gene therapy versus the they both use lentivirus. So, but the difference is the approach with processing the stem cells and preparing the patients to receive the therapy. So the difference is, here's a difference. Number one, the Aruvant is using, initially they were using gamma globin, the hemoglobin A gene, which that is the normal hemoglobin, that's bluebird bio. The Aruvant is now, they're using the gamma globin gene. That's what's used fetal hemoglobin. So they're inserting the gamma globin fetal hemoglobin versus the A. The second is that they use reduced intensity gene therapy. So for example, busulfan is kind of a high dose, if you will, a higher dose intensity treatment to the patient prior to the transplant. The Aruvant gene therapy, 1801, they use a reduced dose chemotherapy. And the reason behind that, the rationale behind it is they want to, that allows them to have less toxicity, right? So if you give more chemotherapy prior to doing the therapy transplant, you can get more infections, you have a lowering of your blood counts, right? So that is that's the two difference. So we call myeloablative approaches. And then also the Aruvant gene therapy probably can be used in more lower resource setting theoretically, because you have less concern about infection, because you're not suppressing the bone marrow to the point where you can get high risk of, you can get infections, right. So that's really the main difference. And in the Aruvant trial, it was for ASH, what they just briefly talked about was interesting. They reported on 3 patients that they're using this reduced intensity trial. And really the bottom line was, it's very early in the process. What they found was that in the first 2 patients, they were treated with the initial lentivirus ARU 1805 with a manufacturing process of the gene therapy, right? But then they presented an improved way of manufacturing, if you will, or optimizing the stem cell collection with the lentivirus production and improved transfer of the stem cells that have the lentivirus into the patient. That's really what they talked about. And what they found essentially was that the first 2 patients who got treated the previous process with this ARU 1804, they fared pretty well. It was a 34-year-old and a 24-year-old that got treated, got transplanted. They got really good early engraftment with the white blood cells and platelets we talked about. But at the same time, we call the fetal hemoglobin that they measured was reasonable. But what they reported was that the third patient had this fetal hemoglobin called G16D, right? With the new processing, they call it process 2 of the stem cells with this new gene therapy. And what they found was they actually had more gene therapy cells into the third patient. They also had they measured more, we call it lentivirus copy number, it's called VCN. That was also found. And what they found also is with this new process, total hemoglobin went from 22% in the first 2 patients that they tried this in, and to as high as 35% in the third patient with the new manufacturing process of the stem cells. So what they found essentially was with this new processing of this patient, the new manufacturing processing of the stem cells with the lentivirus in patient 3 could lead to even 2 to 4 times higher engraftment of the transduced, of the new fetal hemoglobin expressing cells. Number 2, they feel that the reduced intensity holds significant promise because it has a favorable safety profile in patients with severe sickle cell disease. So really, that's really the big difference. It was an early release of their 3 patients with promise of the new processing. That's pretty much it. It's called ARU 1801. So they plan to enroll at least 10 patients, what I saw in the clinicaltrials.gov. So it is promising.
The Bluebird Bio, the new news about the Bluebird Bio that's just come to light. There's so many new and interesting sciences coming. How does that play into where Bluebird Bio is currently, and how other fields and other scientists are utilizing that to further their understanding of this technology?
Yeah, so I think that these 2 abstracts highlight some the very huge upside and promise of this particular type of technology. But as you mentioned, there was some results, recently reported out from Bluebird Bio that there may be some safety concerns that they have identified, and we'll just kind of have to wait and see how that plays out. I'm sure that the company will be very transparent about the findings, and we're all anxiously awaiting those results. But, there are 15, 20 different companies that are looking at multiple different avenues of being able to do this. And for me, that's the promise of this, is that the true curative intent gene therapy is now on the horizon. Like, we have been hearing about it and hoping for it for decades, and it's almost there where you can almost taste it. And for me, that is the long-term promise of this, because hopefully within my career, we'll be able to offer these curative intent therapies to a large number of patients. But for right now, what it means to me is that we need to work with our communities, and we need to work with our patients to ensure that they stay as healthy as we can keep them. As was brought up earlier with, Dr. Wally Smith saying the hum of this disease starts at birth. So we start seeing, not only just the acute complications, but end organ damage very early on in life. And right now, the things that we can do with that are for therapies like hydroxyurea and crizolizumab and some of these other things that we can use to modify the disease.
And that's what's exciting about where we're going with this. This one technology almost gives birth to so much more understanding. And so other companies and other industries are paying attention. So where do they then go from what they're learning? Where do we go from there?
Yeah, so I mean, the hope is that the initial things that we are seeing now are basically just telling us that, yes, this is possible to do. We can do it on a fairly large scale. And we're in the process now of making sure that what we're doing is safe and effective long-term, right? The next steps are to try to be able to do it in as many people as we can if it's safe and effective, which means we have to do it cheaper. We have to do it with less toxicity. And we have to be able to do it in places that maybe not have all of the support of some of the academic hospitals that it's currently happening in.
So who's doing it? Who's already started that process?
So there are certainly collaboratives out there from across academia and through the NIH, the American Society of Hematology, that are focusing on those very issues.
Awesome. Well, guys, that was a fantastic discussion and a wonderful close to part 1 of this series. If you're a warrior listening to this and you think there's somebody who could benefit from hearing what was discussed on this podcast, share it with them. Like and subscribe. Follow me at Dr. Z Sickle Cell, and follow Dr. C at...
And me at
Imagineer. Follow at Cheat Codes Pod on Instagram, and keep living well with sickle cell. That's all we've got for you this week. We'll catch you next time. Peace.